to continue with our discussions, so we have glycine. So the continue discussion with the metabolism of amino acid, So glycine aminotransferase can catalyze the synthesis of glycine from glycosylate, glyosylate, and glutamate or alanine. Okay, so when you say aminotransferase, that is the addition or the transfer of amino group from one structure to another. Okay, unlike most aminotransferase reactions, this strongly favor glycine synthesis process. So additional important mammalian routes of glycine formation or synthesis or production of glycine are from choline and serine. So we have here from serine. So serine, based on this diagram, there is a removal of carboxylic or ester group known as the COOH or carboxylic acid group. So there is what you call a cofactor known as 4-hydrogen and folate. So pwede siya ma-convert ito methylene 4-hydrogen folate, or this reaction is considered to be irreversible reactions. So removal of this one, the decarboxylation process leads to the formation of glycine. Addition of the carboxyl group through the process or carboxylation process of your glycine will form serine. So this is reversible. If you see the arrow, both sides, this is reversible or opposite arrow. So sa reversible process wherein methylene H4 folate is a cofactor is converted to H4 folate, forming now the serine. Okay. Next, we have proline. Proline is formed from glutamate by reversal reactions of the reactions of proline catabolism. So usually we have here glutamate. Glutamate will undergo the dehydration process acted by the reducing agent known as nicotinamide adenine dinucleotide plus hydrogen. It will form L-glutamate semi-aldehyde group, wherein there is a removal of oxygen here. Okay, because of the process of dehydration, removal of water, so one hydrogen is being removed, and the OH, okay, the OH is being removed to form the L-glutamate gamma semi-aldehyde. So there is semi-aldehyde. This is known to be the RCHO functional group. Okay, so again, another dehydration process, removal of water again. So there is what we call a formation of pyrrolidine 5-carboxylate. So the functional group being removed is the aldehyde group. Okay, and then the pyrrolidine 5-carboxylate will undergo another dehydrogenation process. Okay, or what we call this one, removal of the double bond. Okay, acted by enzyme nicotinamide adenine dinucleotide. It will form levo proline. Okay, wherein there is what we call addition. There is a addition of the hydrogen group that is acted by the nicotinamide adenine dinucleotide. So there is a breakdown of the double bond in the imine group. This is the imine group, wherein the hydrogen is coming from NADH will be added to the double bond, which is considered to be the unsaturated bond. Okay, so double bond is considered to be very reactive, and this is what call this one, the structure of pyrrolidine, which is double bond acted by the NADH. So plus hydrogen siya, automatically it will form now proline. Okay, next, cystine. Cystine, while it's not nutritionally essential, is formed from methionine, which is nutritionally essential following the conversion of methionine to homocysteine, and serine form cystine. So based on this diagram, we have homocysteine, okay, and we have the L-serine. So if there is a removal of water, it will form cis cystathionine, okay, removal of water, okay, and then there is what we call addition again of water, then it will lead to formation of L-cystine plus L-homoserine. Okay, yung cystathionine, no, undergoes the hydration process or addition of water to form L-cystine and L-homoserine. Okay, next. By the way, the functional group that are being removed is the OH, okay, from serine, okay, that is bonded by a sulfide bond or linked by a sulfide bond, and then addition of water again. When water is added, it will form L-cystine, okay, wherein there is a bonding of one hydrogen here, okay, and of course another bonding of hydroxyl ion forming L-homoserine. So take note of the functional group. Okay. Next, tyrosine, phenylalanine hydroxylase converts phenylalanine to tyrosine. Okay? So provide that the diet contains adequate, nutritionally essential phenylalanine, tyrosine is nutritionally non-essential, but since the reaction is irreversible, dietary tyrosines cannot replace phenylalanine. Okay? So if you try to see, we have L-phenylalanine, or known to be the levo, okay? So it is converted to tyrosine by hydroxylation process that is acted by enzyme phenylalanine hydroxylase. So there is addition of your OH, wherein your cofactor tetrabiopterin is converted to dihydrobiopterin that is acted by the reducing agent known as nicotinamide adenine dinucleotide plus phosphate, okay, plus hydrogen, then is converted to NADP, wherein 2 hydrogen has been utilized, that is also bonded to that is reacted to water, to oxygen, I mean, so forming your L-tyrosine. Okay, so catalysis by this function, oxygenase incorporates one of the oxygen into phenylalanine and reduces other atom to water. So reducing power provided tetrahydrobiopterin derives ultimately from NADPH. Okay, so the reducing power provided by tetrahydrobiopterin, this is your cofactor, is derived from your reducing agent known as nicotinamide adenine plus dinucleotide plus phosphate plus hydrogen ion. So your tetrahydrobiopterin is known to be the cofactor. Yung reducing agent, tandaan nyo, is the NADH or the NAD. Okay, so take note, functional group that are found in tetrahydrobiopterin is the OH, the methyl group, NH, we have the amino, okay, this is the amide, the amino group. Okay, next, biomedical importance of amino acids. So inborn errors or amino acids in metabolism. So that is why anything that is excess in the amount that is being taken in, I mean, by the human body, so it will lead to certain abnormalities or certain diseases in the body. Okay, so first we have the Phenylketonuria. Okay, so phenylketonuria results from the defect of enzyme phenylalanine hydroxylase, such as phenyl is not converted to tyrosine. So if we have dietary protein, so when we say dietary protein, these are protein coming from our diet. Okay, so phenylalanine and we have tyrosine. So usually phenylalanine is converted to tyrosine that is acted by enzyme phenylalanine hydroxylase. Okay, so in the case of phenylketonuria, there is no conversion of phenylalanine to tyrosine because there is a defect in the enzyme phenylalanine hydroxylase. Okay, And the alternative conversion of phenylalanine is to phenylpyruvic acid, which will be accumulated in the CNS, which can able to cause or can cause severe mental retardations. Kasi when phenylalanine is converted to tyrosine, example, there is no defect in the enzyme PAH or phenylalanine hydroxylase, then tyrosine is converted to melanin that is responsible for the skin pigmentation, proteins, building blocks of the body, and dopamine that is a neurotransmitters which is responsible for cognitive functions, okay, and controls the movement of our body. Okay, so take note of the enzyme that is responsible, or the defect of the enzyme that is responsible for the cause of phenylketonuria. Another one, we have the autoimmune or recessive defect in the reabsorption of the dibasic amino acids ornithine, arginine, lysine from the luminal fluid of the renal proximal tubule and small intestine. So, we will recall our anatomy. So, where can we find the proximal tubule? So, proximal tubule is found near the glomerulus. So, reabsorptions of the different minerals or even drugs, no, after the glomerulus filter, usually it reabsorbs in the proximal tubules. Meaning proximal is nearer to the glomerulus. Okay, so the only phenotype manifestation of cystinuria is cystine urothiasis, so which often reoccurs to affected individuals. Surgical intervention is necessary, but the cornerstone of the treatment is dietary, and medical prevention is recurrent stone. So usually dietary, so we have to avoid eating amino acids like ornithine or arginine and lysine, which is higher in the different food. Okay, so pwede yung surgery kung malaki yung stone dito, pwede kunin yung surgery yan. So urothiasis is known to be the infection or inflammation of the urinary bladder already. Okay, so this is known to be the kidney stone. Next, we have Alkaptonuria or black urine disease, or Alkaptonuria is a rare inherited genetic disorder of phenylalanine and tyrosine. So commonly recommended treatment includes those of amino acids. Okay, so we have here the diagram of the Alkaptonuria diseases. So usually yung mga diseases in the proteins are almost related to each other. Okay, so it is based only in the defect of an enzyme, di ba? So phenylalanine, meaning hydroxylase enzyme, phenylalanine hydroxylase enzyme is an enzyme that converts your phenylalanine to tyrosine. If there is what you call a problem in this enzyme, so it will convert to phenylpyruvic acid, and it will be excreted in urine and also deposited in the CNS and will cause defect. And tyrosine, with the help of enzyme tyrosinase, it is converted to dihydroxyphenylalanine. Then it is converted to melanin. So if there's a problem in tyrosinase enzyme, so it will cause albinism or albino. So makita nyo mga albino, mga puti, no? Usually hindi sila i-expose sa sunlight, ang mga tawag natin mga albino people. Okay, so there is a defect on the tyrosinase enzyme. So tyrosine is converted to hydroxyphenylpyruvic acid. It undergoes tyrosine undergoes the hydroxylation process, and it's undergone another hydroxylation process, then converted to dihydroxyphenylpyruvic acid. And the dihydroxyphenylpyruvic acid undergoes the deoxygenation process, which result in the formation of homogenesic acid. So usually the homogenesic acid is converted to acetoacetic acid, and then it will split into carbon dioxide and water. So this is the normal way, because when it is converted to this one, it will be released in our body. But if there is what we call a problem in the enzyme homogenesic dehydrogenase enzyme, then homogenesic acid is deposited in the body, then it will cause Alkaptonuria. Okay? O ginatawag natin na Alkaptonuria, known as black urine disease or Alkaptonuria. Okay? Next, we have Tyroncinemia. So, in Tyroncinemia, try to see this one. Normally, so we have a source of amino acids, so taken are taken in by the human body. Then all the source of proteins, fish, eggs pa, or meat, then is converted to amino acids during the digestion process. And amino acids, of course, yung tyrosine is converted to fumaric acid and acetoacetic acid with the help of enzyme Fa or or FAH, fumaryl acetoacetate hydrolase enzyme. Okay? While in Tyroncinemia type 1, so same process. So proteins that are taken in by the human body converted into amino acids. Then tyrosine, if there is a problem in the enzyme fumaryl acetoacetate hydrolase enzyme, so tyrosine is not converted to fumaric acid and acetoacetic acid. So there is what we call a building of the succinyl acetone that is converted from tyrosine to succinyl acetone build up, and it will cause health problem already. So this is a genetic diseases. Okay? And there is what we call high level of tyrosine in the blood. Okay? Building blocks of most proteins, di ba? So cause of shortage is one of the enzymes required for the multi-step process of breakdown of tyrosine. That is the tyrosinase enzyme. The fumaryl acetoacetate hydrolase enzyme. So untreated tyrosine byproducts build up tissues and organs, which leads to serious medical problems already. Okay? Next, so treatment, pwede yung diet. So the patient is advised not to eat more amino acid food like meat or egg or chicken, etc. So pwede yung tawag natin na peritoneal dialysis to reduce the level of amino acids. So tama na kadamong amino acids niya, pwede siya natin gawin ng peritoneal dialysis. So long-term treatment requires special diet na. Usually special diet to low level of leucine, isoleucine, okay, valines. So persons with these conditions must remain on this diet permanently. So avoid na lang siya. So, kasi hindi nga ma-digest na ng human body because of the problem of the enzymes fumaryl acetoacetate hydrolase enzyme. Okay? So these are example of the amino acids, okay, the structure. And we go now to proteins. Proteins, the word proteins is derived from the Greek word proteus, means primary. Out of the total body weight, so 3/4 are made up of proteins, di ba? From head to foot, that is made up of proteins. So most content molecules are carbon, hydrogen, oxygen, and nitrogen. Okay, so ano yung mga source ng amino acids? Like of the proteins. Okay, all source of amino acids is also the source of proteins. Okay, so if you try to see the cell structure, this is the bilayer of phospholipids. There are a lot of proteins that is present in the cell surface. Okay, we have the glycoproteins, which is responsible for the transport of glucose, globular proteins for communication process, and protein channels or ion channels are considered to be a protein. Receptors, of course, that are found here are considered to be proteins. All receptors are proteins. Integral proteins or globular for communication process. You have surface proteins also for communication process. And we have integral protein, alpha helix protein. Okay, involved also in the communication processes, peripheral proteins. So there are a lot of proteins that are important in the structure of the plasma membrane or cell membrane. Okay, so ano yung mga protein types? We have structural, enzymes, receptors, and other functions. Using structural proteins, this give shape and structure to the cell organelles is known to be actin and tubulin or myosin. Okay, so usually these are proteins that are found mostly in our muscles. Enzymes catalyze biological reactions. So you have trypsin that is used for that functions for the digestion of food. Trypsin is needed. Adenylate cyclase enzyme for the communication process, or adenylate cyclase enzyme. These are enzymes that converts your adenosine triphosphate to CAM, or cyclic amino phosphate. Okay, CAM. Okay. Next, receptors binds to other molecules for transmit signals. So glutamate receptors, steroid receptor, beta receptors, alpha receptors, nicotinic receptors are examples of receptors. All receptors are proteins. And other, we have antibodies, nuclear factors, and neuropeptides. So neuropeptides are found in the enteric nervous system if you try to remember your anatomy. Okay? So this one, we have amino acids, linkages. In proteins, amino acids are joined by peptide bond. Okay? So, string of amino acids or peptide plus peptide equal form polypeptide. That is a protein. And oligopeptide is a shorter protein or chain which is less 25 chain of amino acids. Okay? So this one, example, peptide bond. So they are being linked by a carboxyl and amino group. So on the negative and the positive side. So this is another one. So linkage by alpha carbon and of course the amino group. So peptides are amide linkages formed by condensation of alpha hydroxyl of one amino acid with alpha amino acids of another amino acid. So this one are example of peptide bond. Okay.